Hi, I'm Annie. Today I'm going to present to you two experiments that ask different research questions. My goal today is to show how research question closely relates to the experimental design, the measurement strategy, and the scientific claim researchers are allowed to make. Before discussing the experiment, I want to define three key concepts. First, correlation. It means two variables vary together. In neuroscience, that usually means neuroactivity is associated with a behavior. However, correlation alone cannot establish causation. Second, necessity. Necessity means without this component, the function cannot occur. We establish necessity by silencing or removing a component. If the behavior disappears, the component was necessary. Third, sufficiency. A neural system is sufficient if artificially activating it produces the function. If activating the component alone produced the behavior, it was sufficient. Purely observational methods like fMRI, EEG, and other neural imaging can establish correlation alone, but causation claims requires manipulation, such as by using lesions, electrical stimulation, etc. This brings us to my research questions. The first study asks a correlational question. Is hippocampal neural activity during post-learning sleep associated with the strength of subsequent memory recall? Because the researchers only observed the brain activity using fMRI, this is a correlational design. The second study asks a causal question. Is hippocampal neural activity during post-learning sleep causally required for memory consolidation to occur? Here, because we experimentally manipulate or inhibit the network using rTMS, this is a necessity design capable of supporting causal inference. The research question determines the method. Study 1 used a within-subject correlational design. Participants first complete an encoding phase where they learn paired words from two separate lists. List 1 was learned first, followed by list 2 1 hour later. After encoding, participants entered a retention interval. Depending on the condition, this interval included either sleep or wakefulness. Finally, participants completed a retrieval test in which they attempted to recall the correct word pair associated from both lists. Throughout the experiment, researchers measured hippocampal activity during encoding using fMRI. Functional fMRI measures the bold signal, which reflects the change in blood oxygenation associated with neuroactivity. Importantly, researchers only observed hippocampal activity. They did not manipulate the memory system. This keeps the design purely correlational. Several experimental controls strengthen the internal validity. First, participants are unaware that a subsequent memory test will take place on day 2. This prevents their conscious rehearsal during waking hours. Second, the within-subject design allowed each participant to serve as their own control, reducing variability from individual differences in baseline memory ability. Third, the design included a baseline condition without contextual information, helping to isolate hippocampal activity specifically related to contextual memory encoding. Study 1 established a predictive relationship between hippocampal activity and memory performance. However, prediction alone cannot establish necessity. Memory consolidation occurs primarily through non-REM sleep, when hippocampal network replay recent experiences. This leads to next question. If we disrupt hippocampal network activity during sleep, does memory consolidation fail? Answering that question requires experimental manipulation. Study 2 uses a within-subject crossover design to test necessity. Participants first learn word-image pairs and then complete an immediate recall test before sleep. This established a baseline measure of encoding performance. Next, researchers applied repetitive transcranial magnetic stimulation, or rTMS. This is a non-invasive method that uses magnetic pulses to temporarily suppress activity in a targeted brain region. We target the lateral parietal cortex, a cortical region with direct functional connectivity to the hippocampus. This transiently suppresses the hippocampal network activity during early non-REM consolidation window. Unlike fMRI, which measures activity, rTMS actively manipulates the neural system. This is why it can support causal inference. Participants then sleep overnight and complete a memory test the next morning. The dependent variable in study 2 is overnight memory retention, calculated as memory recall minus pre-sleep recall. Researchers compared memory performance before sleep and after sleep. Then they compared two conditions, active rTMS and sham rTMS. If memory retention becomes worse after active stimulation, this suggests hippocampal network activity during sleep was necessary for consolidation. The study includes several important experimental controls. First, the sham stimulation condition makes the participant experience similar sounds without neural disruption. This controls for placebo and expectancy effects. Second, the researchers used a counterbalance within subject crossover design. Every participant completed both active and sham session in different orders. Counterbalancing control for order effects, while the within subject structure reduces variability from individual memory differences. Finally, sleep was monitored using polysomnography, including EEG recordings, etc. This ensures that any memory differences are not simply caused by disrupted sleep architecture. Together, these controls strengthen the causal interpretation of the findings. To conclude, across both studies, the central principle is that research question determines the research experimental design. Study 1 uses observational method to test correlation. Study 2 uses manipulation to test necessity. This distinction is important in neuroscience because method determines the type of conclusion we can make. Observational study can identify relationships. Manipulation is required to establish causation. Thank you.